Researchers in France have reported the emergence of a new SARS-CoV-2 variant, currently designated B16402, from the analysis of 12 SARS-CoV-2 positive patients living in the same geographical area of southeastern France. The research paper, which is yet to be peer-reviewed, has given rise to fresh concerns on social media at a time when India is already facing a third wave of COVID-19, with the Omicron variant spreading fast. The researchers are calling it the IHU variant, although research groups are not authorized to name variants. Variants are designated names by WHO if they are of concern. I am Mohana Basu, and in this video I take you through everything we know so far about this variant. The study describes that the first patient identified to be infected with the variant had returned to France from a trip to Cameroon. Every living organism is run by a predefined set of genes that instruct cells how to build proteins. Understanding the sequence of genes is thus like cracking the code to the organism and how it functions. The genetic material of the coronavirus is ribonucleic acid, or RNA strands. Each virus has about 26,000 to 32,000 bases, or RNA letters, in its length. These letters A, C, U, and G stand for adenine, cytosine, uracil, and guanine, which are nitrogen-containing biological molecules that are the fundamental units of the genetic code. How A, C, U, and G are arranged in the genetic code determines what amino acids are expressed by the organism. Amino acids are the building blocks of proteins. When a virus multiplies inside the cells of a living organism, it creates copies of the RNA. However, the process it uses to make these copies is not perfect and often introduces tiny errors in the sequence of bases. These errors are called genetic mutations. A deletion is when one letter gets missed instead of getting replaced. The team in France found at least 46 mutations and 37 deletions in the new variant compared to the original viral strains. This resulted in 30 amino acid substitutions and 12 deletions. According to the researchers, the data is another example of the unpredictability of the emergence of SARS-CoV-2 variants and of their introduction in a given geographical area from abroad. Vinod Skaria, a researcher at CSIR who has been tracking COVID variants and mutations, wrote in a Twitter post that given what is currently known about the variant so far, there is no need to panic. The variant has been on the WHO watch list since before the emergence of Omicron, Skaria said, and its sequences have not grown rapidly. B1640, the parent lineage from which this new variant has emerged, was detected in November last year in Ghana and is being monitored since. Less than 400 samples of this variant has been reported across the world so far. Some of the mutations on the spike protein of this variant are the same as those identified in past variants of concern. These include N501Y and E484K, both of which have been associated with the ability of the virus to escape the body's natural or vaccine-induced antibody response. The researchers from France also pointed out that, like the Omicron variant, the mutations on the spike protein of this variant make it easy to detect using RT-PCR test. This means that the variant can be easily screened with certain RT-PCR tests without the need for a full genome sequencing, which can take a much longer time. This is Mohana Basu, special correspondent at ThePrint. Do follow us on social media platforms for the latest news updates.